So today I get to talk about one of the cooler molecules in our body, and that is glycogen. So let's just jump straight into it. Now, I've talked previously about how a muscle cell might be consuming energy, and let's say it's burning glucose. It's getting its glucose from a couple of different places. One of those places is its internal glycogen stores, the topic of today's video. And then the other is from the bloodstream. But it turns out that glucose from the bloodstream also probably was glycogen at some point, because as blood sugar drops, the pancreas releases the hormone glucagon. Glucagon signals to the liver, hey, start breaking down your glycogen stores and dumping them into the bloodstream, thus maintaining blood sugar at a particular level. So glycogen plays this important role both as a local fuel source for muscle cells and as a global fuel source for balancing blood sugar. And we can actually show this explicitly with a very personal example. And this is from a very long run that I went on to, where I didn't actually eat enough food. And so we've got time on this axis, my heart rate on this axis, and hopefully what you can see is right here is where I ran out of glycogen. And it turned out it sucks, right? Your heart rate spikes because you have to switch over to a different fuel system. If you care about things like pace, your pace falls apart. You get very tired. So glycogen is really important for maintaining our blood sugar and also for allowing us to do things over metabolic time. Doing metabolic things over time. There we go. Just to orient you in the transit map of where we are, this is what we've talked about to this point in these videos. We've talked about glycolysis, which is sort of going from glucose down to pyruvate. We've also talked about lactate fermentation over here. And we're going to visit this set of maps, or this set of routes here, going in and out of glycogen. At the very top of glycolysis, this feeds straight into those energy production pathways. So if we look at all the different things that routes that we have and access to in the central carbon metabolism, we're going to be talking a lot today about these two processes: glycogenolysis, so breaking down glycogen, making glucose, and glycogenesis, storing glucose as glycogen. So we're just going to go through this molecule with a set of questions. First, what is glycogen? And glycogen is a highly branched polymer of glucose, and this is shown in sort of somewhere between a real picture and a cartoon here on the right, where we've got a molecule of glycogen in the middle. This is a protein that's sort of a nucleus for glycogen. And we have these branched patterns of glucose coming off them. It's found primarily in the liver and skeletal muscles. It's like I showed in the earlier cartoon. It's actually found in small quantities in other cell types. But these are the big producers and consumers of glycogen. If you look at a hepatocyte, you can actually see glycogen. So hopefully, if you're looking at these, you see all these black granular things. Those are glycogens that are packed tightly inside of the cell, right? So it's, like I said, it's this highly branched network, this polymer of glucose. It's so dense and packed that you can actually see it on an electron microscope as a dark blob, basically. What's so cool about glycogen? There's a bunch of things that are cool about it. One of my favorites is that it actually pumps sugar into the cell by Le Chatelier's principle. So here I'm drawing schematically blood at the outside, cell on the inside. Let's imagine for a moment that our cells are at equilibrium. We've got an equal concentration of glucose in the blood relative to what's in the cell. But then we start doing polymerization. We start making something like glycogen. We're polymerizing that sugar. Now what we've done is we have depleted glucose in the cell, and so glucose diffuses in and polymerizes. So basically, even though we're not directly pumping glucose, there's no pumping mechanism that's dragging things in, we are pumping glucose in because we're accumulating it in these glycogen polymers that basically store the glucose and pull it out of the blood. Same thing happens in reverse, actually, right? So now let's imagine we've got low blood glucose. We've got a bunch of polymers of glycogen inside, say, a liver cell. If you start depolymerizing, just naturally what's going to happen is you're going to start seeing diffusion out, right? So it's a really nice way to pull glucose into a cell, say a liver cell, and release it just by making the polymer. You don't have to do some complicated export process. Another cool thing about glycogen is I said it's a branched polymer that allows for fast sugar access. So if we imagine the way that we get to this is we chop each glucose off one by one. This is this array of glucoses. If we did that for one polymer, We can do it. But if we make a branched polymer, now we've got 1, 2, 3, 4, and behind me, 5 different ways that we can access it. So we can basically rapidly expose and release a bunch of sugar just by adding a phosphorylase. It's going to chop those off. And finally, the branching pattern actually increases the glycogen and thus the energy density. And it's not quite the right picture, but I kind of like it. Just imagine grass, right, is fairly unbranched. If you sort of pack grass together, it's sort of loosely packed unless you apply external pressure to pack it together. Whereas you think about something like broccoli, where it's got this branching pattern, it gets this really dense packing, right, because you're branching and articulating. We sort of see the same thing with glycogen, where because it branches, it can sort of fill in the niches and make this really dense material that's storing a lot of energy in a small space. And that's why when you look at it from in one of these electron micrographs, you can actually see, oh yeah, like there's a bunch of glycogen because of this high density of packing. Okay, so glycogen is this cool polymer. How is it synthesized? And there's a 4-step synthesis process that I'm going to describe really quickly, and then we're going to go through the steps one by one. But first, we're going to prime glucose subunits by attaching a UDP at position 1. We're going to build a short chain of sugars on the protein glycogenin. There's a word I just cannot say. Glycogenin. We grow existing sugar chains that are nucleated on that protein in something called alpha-1,4 linkages. And then we finally break off those 6 to 8 subunit chunks and append them to build the branch. So let's just go through these really quickly. Priming that. That is not, you want to see the slides, not me. Okay, so yeah, see me and the slides. We can do this. Okay, so quick reminder, in case it isn't resident in your memory, that glucose is a 6-carbon sugar whose carbons are numbered 1 through 6. And so in the open form here, we would number it from the top 1, 2, 3, 4, 5, 6. And then in the ring form, where this is closed to form the favored Pyranose form, the 1 carbon is traditionally drawn here, and it's always next to this hydroxyl in the ring. And then 1, 2, 3, 4, 5, 6, 6 is drawn in the back there. And so when we're talking about linkages, what we're going to be referring to are which carbons we're talking about. So that's where the 1s and the 6s and the like come from. Okay. So let's walk through the steps. Remember, the first step here is we're going to prime the glucose with UDP to be put into glycogen. So the first step in this is actually one we've seen before in videos on glycolysis, right? The first step in glycolysis is adding a phosphate at position 6 to glucose. So glucose to glucose 6-phosphate. We place the phosphate there on that 6th position. Same enzyme is involved in glycogen synthesis. After we put that phosphate on, we then do a process called, or an isomerization process. We put the phosphate onto the 1 carbon, catalyzed by the enzyme phosphoglucomutase. And then we have a 2-step process, which I'm going to show schematically, and then I'm going to show with molecules. We're going to take a UGPase, and we're going to use that to take a UTP and make a glucose UDP that releases this pyrophosphate. That pyrophosphate then gets cleaved by pyrophosphatase into 2 inorganic phosphates. So that's a lot of stuff happening there, so let me unpack that really quickly on a slide. So here we have our G1P, and here we have our UTP. UTP is exactly like ATP, but instead of having an adenine base, we have a uracil base. What is going to happen in this reaction is we are going to basically have a nucleophilic attack by the phosphate of G1P on this phosphate here. That's going to break this bond, and we're going to be left with the inorganic phosphate, the pyrophosphate as it's called, and then this is our glucose UDP, right? So it's got 2 phosphates and then that UDP base. And the reason that this is an important step is because we have just made like the world's best leaving group right here, right? So that bond is primed and ready to break because there's all kinds of interesting ways to move the negative charge across these 2 phosphates. It's big and bulky. This thing just really wants to fall off, so it's a good leaving group. So this is primed to be put into the polymer. This by itself actually is approximately reversible. We just swapped phosphodiester bonds for other phosphodiester bonds, so there's not a lot of energy to be had here to drive this reaction forward. Hence the next reaction, and that is driven. This is pyrophosphatase, is actually cleaved into two inorganic phosphates. That is extraordinarily energetically favorable to do. We're breaking a phosphodiester bond. And so that is the thing that drives the reaction forward. So this first bit just happens. It's relatively energetically neutral. This is energetically favorable, providing the driving force. And it turns out that that phenomena of having a pyrophosphate be cleaved into two inorganic phosphates comes often, and it's actually a common biosynthetic mechanism. We see this across different biosynthetic pathways where you form the pyrophosphate, cleave it in half, boom, you've got driving force for a reaction. Okay, so we just primed the glucose subunits by attaching a UDP at position 1. Now we're going to build a short chain of sugars on this source protein, glycogen. There we go. And glycogen is a really cool little enzyme. And what it does is it catalyzes building sugar chains. So it'll take these glucose UDPs and it will start polymerizing them, but it does it on itself. And so hopefully you can see there, it's a little bit, I'm going to zoom in so we can look at it. There's a tyrosine residue that's hanging out right off of the active site. And this crystal structure was solved after this is already polymerized. It's put on 4 different sugars. Those 4 sugars just get appended on. So it's building out an array of glucose molecules that is going to nucleate or form the basis for a new glycogen array. Okay. All right. So we know how we're priming our sugars. We know how to start building the chain on this thing glycogen. I need to, anyway. Now we're going to grow the existing chain alpha 1,4. And so what we're going to do here is, now I'm drawing this schematically. We've started out with our glucose UDP. We're going to use the enzyme glycogen synthase to append a glucose onto an existing chain. So that is shown here. Let's look at how that works molecularly. So what we're going to see here is, remember I said that this is now a really good leaving group. What that means is that the oxygens of the 4-hydroxyl can attack this carbon, and this will happily, will happily knock off the UDP. What that yields us is a UDP and another subunit added onto there. So we've gone from 4 now to 5. And this reaction happens spontaneously because this leaving group is so good at leaving. Okay, so what we have here is a glycogen chain. I'm showing here schematically. We've got in red and blue, these are 1,4 linkages. And what glycogen synthase does is it just continues on. It'll just keep sticking these on, making the chain longer and longer and longer. And the way it's growing is it's actually growing this direction on this picture. Okay, so we've primed sugars, we've started chains with glycogen in, now we are growing the chains. How do we get the branching? And this is just mind-blowingly cool. So just to make sure we're on the same page about what the branches are, so these are main chain. These are alpha 1,4 linkages. We know they're 1,4 because they've got a 1 and they've got a 4. That's where the 1,4 notation comes from. And in case you want to know, they're alpha because this oxygen is below the ring. It would be beta if it was above the ring. A 1,6 linkage is you take the 1 of one glucose and you link it to the 6 carbon of another glucose. And again, it's alpha because this is below the ring. So glycogen consists basically of alpha 1,4 linkages and alpha 1,6 linkages arranged in an interesting way. So how does this work? There's this enzyme called glycogen branching enzyme. And what it does is it builds out the branch structure. So glycogen synthase sticks out a bunch of glucose molecules into an array. The glycogen branching enzyme recognizes long stretches of about 10 or more subunits that are linear, and it chops off 6 to 8 subunit chunks. Those are now separate from the rest of the chain. It then identifies gaps of 3 to 4 polymer subunits, and it fuses that chain on. So now we've built a branch. Glycogen synthase is still doing its thing, right? So it's going to be growing this chain, it's going to be growing this chain, and it's going to continue to grow this chain. And so by the combination of these two enzymes, the synthase driving out making more chains and the branching enzyme branching, you build out this dense branch network with highly accessible glucose. Okay, so pretty freaking cool molecule, I think. Very briefly, let's talk about how glycogen is broken down. And so it's broken down using an enzyme glycogen phosphorylase. And what glycogen phosphorylase does is it chops this molecule off and regenerates G1P. So that's totally ready to go rock on and do the rest of metabolism. The way that this works sort of phenomenologically is glycogen phosphorylase chews back linear chains, but it can only chew back to within about 4 subunits of a branch point. So at this point, it's stuck. It can't chew further. So it brings in a friend, the glycogen debranching enzyme. And what the glycogen debranching enzyme does is it chops off 3 of them, glues those onto the end of an existing chain, and then removes the final one. And this is actually a pure glucose molecule. It's ready to go off and be used immediately. It doesn't even have a phosphate on it. And then these molecules here get chewed back, or these subunits, by glycogen phosphorylase. So the collaboration of glycogen phosphorylase and the glycogen debranching enzyme breaks down the chain, or breaks down the branching pattern. Glycogen synthase and glycogen branching enzyme build out the pattern. Okay, last question, then we'll be done with this video, is what controls glycogen formation and breakdown? And hopefully what you're seeing there is you're like, I see some phosphorylation-y bits. And maybe I've even seen those before. If you've watched earlier videos of mine about sort of fuel, carbon fuel sort of reciprocal regulation, turns out that these phosphorylation reciprocally regulates this process. And so this small GYS, glycogen synthase, when it is dephosphorylated, it's active. And when it's phosphorylated, it's not active. In contrast, the glycogen phosphorylase is inactive when it is dephosphorylated, and it is active when it's phosphorylated. So if you can phosphorylate the molecules, you switch over into glycogen breakdown. And if you dephosphorylate the molecules, you switch over into glycogen formation. Where do we get that signal from? Turns out we can get that all the way up here from hormone signaling, right? And so what I'm showing here is glucagon signaling can go, and glucagon signaling activates this phosphorylation, which drives glycogen breakdown. Insulin signaling comes over here. It dephosphorylates and drives glycogen synthesis. So we get this reciprocal regulation that's hormonally regulated just by regulating the activities of glycogen phosphorylase and glycogen synthase. And what this then allows is for this coordinated regulation, right? When blood glucagon goes up, that says blood sugar is low. That says that cells are consuming glucose from the blood. That triggers the breakdown of glycogen to form glucose. When blood glucose is high, insulin gets released. Insulin triggers the formation of glycogen, which actually leads to a net import of glucose into the cell, and thus lowers the blood glucose. Okay, so what have we said in this video? I've said that glycogen is a highly branched polymer of glucose that's used for energy storage. The branching pattern allows for really dense packing in space and a rapid release of energy. And that the synthesis and breakdown of this are coordinated by phosphorylation of glycogen synthase and glycogen phosphorylase, which is ultimately linked all the way back into glucagon and insulin and hormonal signaling in the bloodstream. So that is all I have on glycogen. I hope it was interesting, and I look forward to seeing you later.